here's how it's different than the others?
Yes, no, so as far as, yeah, like the biochemical pathways, I would know what the main metabolic pathways are: glycolysis, citric acid cycle, pentose phosphate pathway, wow, Calvin cycle, fermentation, all those. Know what it starts with, what the product is, and why like that product's important for the cell. And the energy state of the cell, right? So like glycolysis and the TCA, the purpose is to generate, the reducing agents for the electron transport chain, which is where you get the bang for your buck for ATP. And then the pentose phosphate pathway is anabolic. So if the energy state is high enough in the cell, then you can use energy to synthesize like nucleic acids and amino acids, like all the biosynthetic stuff. So yeah, that kind of big picture.
Okay, but we don't need to know the in-depth, like each step in those processes.
No, I don't think so. Mm-mm. Knowing like, here, let me see if I can share the PowerPoint, Sarah. Knowing like the transition, like the overall picture, like the steps, like the transition step from glycolysis to the TCA cycle. I think one of the questions is like, why is coenzyme A important? So, and that's to get acetyl-CoA into the TCA cycle. Let's see. Those chapters, this chapter 6 maybe. Yeah, and I would know how much ATP is generated from glycolysis. So you get 2 from glycolysis and 2 NADH. Ooh, so pentose phosphate pathway. It does break down glucose, but it generates NADPH, and that's an anabolic reducing agent. Yeah. So yeah, Sarah, you're right. I don't think you're gonna have to know all of the enzyme names and stuff. I think I would know glucose 6-phosphate feeds into pentose phosphate pathway or glycolysis, because that's activated glucose. And then I think I would also know glyceraldehyde 3-phosphate feeds back into glycolysis.
Okay.
If he has it listed on a slide, I would know the name. But like some of the, pathway figures and stuff, like you're not going to have to know every one of these enzyme names.
Okay. And then the pentose phosphate pathway, that's the only one that makes the NADPH, right?
Yes. Yes. And you've got the 2 phases: oxidative that generates NADPH, and the non-oxidative one generates the 5 carbon sugars. And you think about those are sugars that are like needed for DNA synthesis. Yes, and glyceraldehyde 3-phosphate and fructose 6-phosphate, those can feed back into glycolysis. They're all connected. Like I could envision, like I don't know what questions he'll actually put on there, but like just a general essay of like what are the big metabolic pathways in a cell and describe them,
Okay.
Yes, you're right, Sarah. The only source of NADPH, reducing biosynthetic reducing power. And oh, I would know that it's regulated by glucose 6-phosphate dehydrogenase.
Okay.
So yeah, any enzyme that he has listed on here that he highlighted, I would know it. And then yeah, the transition step that connects glycolysis and the pentose phosphate pathway to the TCA cycle. Yep, the transition step is with coenzyme A.
And what does coenzyme exactly?
What, Sarah?
For coenzyme A, how does that play into it? Or do we just need to know that it does?
I would say, let me find a picture. That's glycolysis. CoA is what, let's see. Yeah, CoA is what oxidizes pyruvate to acetyl-CoA. And so CoA, when it attaches to this 2-carbon acetyl group, it becomes acetyl-CoA, and that's what lets the carbon get into the TCA cycle. Right.
It binds to half of the pyruvate.
Yes. So pyruvate, you lose a CO2, and then you have an acetyl group, and then coenzyme A binds, and then you have acetyl-CoA right here. And this is part of the transition step. So that's probably a keyword to note that connects glycolysis and the pentose phosphate pathway to the TCA cycle. And for every pyruvate, you get 2 acetyl-CoA.
Yeah.
So you would technically have, I guess, 4 of these, right? Because glucose gets split into 2 pyruvate.
Yes. So then for the NADH, do you get 2 total or 4 total?
In the TCA for ATP.
Transition step.
Oh, for the transition step. Let's see.
That slide right there. The way, yeah, because the way it's written, it looks like it might be 2 NADH per single pyruvate, but I don't know if that's how he meant for it to come across.
Okay. For pyruvate, you get 2 acetyl-CoA. Okay, yeah. So this would happen twice. I see what you're saying. For each pyruvate.
Would it happen twice for each pyruvate or just once? That's the part I'm confused about.
Yes. It would just happen once for pyruvate, right? Because you're losing this CO2 here at the top. Can you see my cursor moving?
Yeah.
Okay. So you would lose this CO2, have your acetyl group, coenzyme A would bind, and then there's a thiol that forms. Yeah, and that only happens once per pyruvate. Oh, so maybe he's saying, so I guess it would be 2 pyruvate that goes into 2 acetyl-CoA.
Because I think it's 2 acetyl-CoA per pyruvate. The NADH part was confusing to me.
It is 2 acetyl-CoA per pyruvate.
Pretty sure. That's what I have in my notes. But I was also a little confused during the lecture.
Yeah. Let me see. Let me Google it really fast. Okay. The 2 pyruvates that enter glycolysis for each glucose molecule, we can summarize pyruvate oxidation as follows. 2 molecules of pyruvate are converted into 2 molecules of acetyl-CoA.
Okay. So that would just be 2 NADH then.
Yeah, and he has that here. Oh, so I remember in class he said that this needed to be 2 pyruvate.
Okay.
Do you remember that?
Kind of.
That was so long ago.
I haven't had cell bio yet, and so a lot of the stuff from this unit is just like a lot to take in.
Yeah. I totally get that. But yeah, I think we got that. But yeah, just knowing that coenzyme A is important for the transition step to oxidize pyruvate into acetyl-CoA, and then acetyl-CoA is what enters the TCA cycle in the mitochondria. Maybe there's... Woohoo! Yeah. Dude, I want that top part to go away. Okay. Yeah, so pyruvate gets converted into acetyl-CoA that enters the citric acid cycle. Yeah, and this is showing one pyruvate, one acetyl-CoA, but then there would be two of these from one molecule of glucose.
Okay.
And then the TCA cycle is all about generating these reducing agents that then go to the electron transport chain, donate electrons, and then you have electron carriers, and then so much ATP from the electron transport chain.
So all of the other path, like not the electron transport chain, but everything leading up to it is just making the reducing power for the electron transport chain.
Or biosynthetic pathways. That would be the PPP.
Okay.
Yeah, here's a good overview.
So definitely knowing this chart would be useful while knowing like what the precursor.
Yeah, I was getting ready to say I would know the pentose phosphate precursors, the transition precursor, which one precursor metabolite? What?
That's the pyruvate.
Pyruvate? Okay. Yeah.
Pretty sure.
What happened? Yes. Yes, I think you're right. And there can be 6 different precursor ones for glycolysis. Well, glycolysis generates 6 different precursor metabolites. Let's go back.
So for the transition step, that would be acetyl-CoA.
Yes, yes. So it said for glycolysis, it generates 6. So yeah, here's glucose 6-phosphate, fructose 6-phosphate, dihydroxyacetone phosphate, 2-phosphoglycerate, phosphoenol pyruvate, and pyruvate. 1, 2, 3, 4, 5, 6. Yeah. But I mean, you're not gonna, I don't think you'd have to know all of those. Definitely pyruvate.
Yeah. I remember talking about pyruvate and then the glucose and fructose. I don't remember talking about the others.
Yes, I would say knowing glucose 6-phosphate, it's just a phosphorylated glucose and it's converted into fructose 6-phosphate. Yeah. And then knowing the total ATP yields from each of them. Yeah, and here's the 2 precursors for the PPP, ribose 5-phosphate for nucleic acids and erythrose 4-phosphate for amino acids. I don't know if you would get that specific. But yeah, and your oxidative phase of PPP generates NADPH, and your non-oxidative generates your 5-carbon sugars.
Okay.
So yeah, knowing all of the products from each of these pathways would be good. And the non-oxidative, you also get some G3P and some fructose 6-phosphate that can feed back into glycolysis.
And then for smallpox, it shouldn't be too difficult of questions, right?
Yeah, I don't think so. Let's see. Let's see. He said up until this, right?
Yes.
Yeah, because he showed those pictures. Yeah, so knowing the differences between the types of smallpox, like malignant is flat, it's nearly always fatal. Hemorrhagic is always fatal, usually massive bleeding. Ordinary and modified are not usually fatal.
So ordinary would just be like your generic smallpox.
Yeah. Like we always hear about. Yeah, the typical rash. But there's more on his little face. Or she. Ooh, and maybe knowing the progression of smallpox.
So for the progression, what are papules?
What are papules?
Because that kind of skips over them, but they're in the previous slide.
Here, let me Google it real fast. It sounds like they just might be bigger pus. No, well, there's pustule. Let's see. Papules, smallpox.
Because if the macules are
Okay.
the red spots,
Wait,
and
go ahead.
vesicles are clear fluid filled.
The pustule.
Yeah.
Yeah. It says initially smallpox lesions are small papules, but they change into vesicles and pustules in 1 to 2 days. So the papules are not fluid filled, but the vesicles and pustules are.
So are the papules just a bigger version of the macules?
I think so.
Okay.
Yeah. And that's something so so detailed. But still good to know. Enanthem, that seems like a very random word. Lesions in mucous membrane of the mouth and nose. Because, like, for his fill in the blanks, they're going to be kind of random. Not random, but specific. And your most, okay, yeah. And so if you go in the PowerPoint and look at his notes under each slide, that's a good way to study too. So the most contagious, you're most contagious in this phase. When you have lesions in your mouth and the macule phase.
So are the lesions in the mouth during the macule phase? Or are they just kind of whenever?
Yes. I would say so because it's under the macular one. So I think I would say I would feel good saying that. You feel good about smallpox?
Yeah.
Okay.
Do you think he'll ask about the history since he spent quite a bit of time on it in class? Or
I don't remember him asking us a ton about the history of smallpox. Maybe a little bit about like the variolation and vaccination. That variolation means grinding up scabs, and then they would inhale it. That seems like a good vocabulary word. And then, yeah, I don't think you're gonna have to know the genus and species of it.
would knowing that like cowpox and monkeypox are a thing and that they were used originally in vaccines help?
Yeah.
Because you did mention that originally it was cowpox. in the vaccine.
Yeah. Yeah. That's also a cool fun fact to know. But yes, that's good. Oh, okay. Well, this isn't that hard to kind of memorize. Variola only infects humans. That's major problem. That's the deadly form, and variola minor is the mild form of smallpox. So it's honestly, I looked at it and I was like, ooh, that looks hard, but it's not that hard.
So is orthopox just like the category that all of the different types of pox fall under?
That's what I was trying to think about. Let me see. I think so. Orthopox. Orthopox virus is a genus of viruses. Is in the family of Poxviridae, so pox viruses, I guess. Vertebrates, including mammals and humans, serve as hosts. Oh, okay. So I guess variola is the only one that infects humans. But then cowpox affects cows, monkeypox affects monkeys, and then vaccinia is used to make the vaccine and can infect multiple species of mammals and natural hosts. But variola is the only one that infects humans. So orthopox encompasses all of the genuses.
Okay.
Yeah. Cool. Good. Is there anything else specific, Sarah, that you can think of?
I think that's it.
Cool beans.
Thank
Do you
you.
want to quickly go through anything else?
No, I think I'm good. I just need to sit down tomorrow and go through most of the diagrams.
Yeah, that's a good way to study for sure. I might stay on here and record going through the review questions. Just really fast.
Well, thank you for hosting the review sessions this semester. They've been helpful.
You're so welcome. A lot of people said they couldn't come tonight, so I didn't know if anyone would be here live.
Yeah, there's a biochem exam Friday, so a lot of people are studying for that.
Right, and then taking it on Monday. You're right. That's true. Hey, good luck in all of your stuff, Sarah.
Thank you. You too.
Thanks. Bye.
Bye.
Okay, friends. I will go through some of these review questions and just point you guys to places to review because I know you guys have a lot to do. We all do, but we can do it. So let's see. Let me share here. I'm just going to start with genetics because it's my favorite. Okay, so I'm going to just kind of go through his review questions that he put on Moodle. So his first question is describe DNA replication. So let's see if there's a slide. So DNA replication, you guys know that there's an origin of replication. Bacteria, they only have one chromosome, so they only have one origin of replication, right? And plasmids, the little extra chromosomal DNA that bacteria can have. In eukaryotic cells, we have very large chromosomes and they're linear, so there are multiple origins of replication on each eukaryotic chromosome. And it's bidirectional replication, so there's the replication fork. Oh, here's a picture. Oh, that's in bacteria. Okay, here we go. Yeah, the replication forks, you start at the origin and you start synthesizing DNA in both directions because it's faster. And in eukaryotes, it's the same thing. And you separate the strands with helicase, they bind to the origin and unwind the DNA because you need DNA polymerase, the enzyme that actually synthesizes the DNA strand, needs to access the DNA template to read it, right? And once the DNA is unwound, you actually have primers synthesized first because that's the starting point for polymerase. It needs a place to prime off of, right? And again, like go through the PowerPoint and read his notes down here at the bottom too. That'd be a good way to study. So yeah, DNA polymerase, it synthesizes 5' to 3'. The nucleotides are added on to the expanding 3' strand, and you can only add to pre-existing nucleotides, right? Because you so it synthesizes 5' to 3'. The 3' hydroxyl is a starting point. The 3' OH on the sugar is where polymerase recognizes and can start adding on new nucleotides, right? Ooh, and here's a good picture of the replication fork. Yeah, as long as you know, you don't have to know, I think, all the details of this process. I think I remember him saying that in class, like he's not going to ask you all of the proteins and the steps of DNA replication, right? So DNA replication, really the purpose is to just copy the DNA, right? And knowing that it's faster in prokaryotes, there's only one chromosome. It is a big chromosome though. But yeah, and knowing that they only have one origin. And then transcription, is to synthesize an RNA transcript, and you've got messenger RNA, ribosomal RNA, and tRNAs. And tRNAs are what aid in translation, bringing in the correct amino acid. And one more big picture thing, like you guys know, is that replication, transcription, translation, it all happens at the same time, like in the same place, because there's no nucleus, right? So in transcription for bacteria, you have sigma factor and RNA polymerase. They associate together and then scan along the DNA, and then sigma factor recognizes a promoter. And a promoter is where transcription begins, right? And then, like in DNA replication, the strands are separated, and then RNA polymerase transcribes the DNA into RNA. Yeah, and here's a good summary chart, but you guys are good with that. Yeah, so sigma factor helps with transcription initiation, and sigma factor is specific to bacteria. In eukaryotes, in us, we have a similar kind of process, but we use transcription factors, a lot of different transcription factors. In bacteria, it's just a little simpler, just sigma factor. Ooh, and here's another good difference. I think one of the questions was, what are the differences and similarities in replication, translation, and transcription between prokaryotes and eukaryotes? So this is a good difference. In prokaryotes, you can have a monocistronic or polycistronic sequence. So polycistronic, that's like when bacteria have their genes organized in an operon so that they can transcribe multiple genes under the control of the same promoter. In eukaryotes, most genes are one gene, one promoter. and a sequence that is one gene, one protein, right? So that's a monocistronic. And polycistronic, multiple genes and multiple proteins synthesized or transcribed from the same, under control of the same promoter, organized in an operon, and they usually have related function, right? So the lac operon, those genes are involved in metabolizing lactose. So it makes sense for the bacteria, if lactose is present, then just transcribe all the genes you need at once to metabolize lactose. Cool. Then translation, you have your messenger RNA that's going to be translated into the protein, and the messenger RNA is read by the ribosome, and the amino acids are brought into the ribosome by tRNAs. Yeah, and the genetic code is read in sets of 3. The start is AUG. All of them start with methionine. And then the stops are like UAA, UAG, UGA. Don't think you're going to have to know these specifically. And the code is degenerate, right? So more than one codon encodes the same amino acid. Yep, in your tRNA, it has an anticodon sequence that's complementary to the codon sequence, and that what brings in its amino acid specifically. Each tRNA is charged with its specific amino acid that corresponds to its anticodon. Yep, and there's a fancy enzyme that charges tRNAs with their amino acids. It's called aminoacyl tRNA synthetase. It's an ACE, so it's an enzyme. And there's a tRNA synthetase for each of the amino acids. So there's an alanine acyl, or a glutaacyl. Not sure what they are actually called, but there's one for every amino acid, because you need to charge each tRNA with its specific amino acid based on whatever anticodon sequence it has. The enzyme recognizes the anticodon sequence, and the appropriate amino acid is put onto the tRNA. And again, I don't think you'll need to know the ins and outs of these steps, but just knowing like big picture, translation translates the mRNA into a protein through the ribosome, and the ribosome brings in the right amino acids, right? So let's just focus on this. Well, yeah, again, read his notes down here to help you. Whoop. The start codon first starts out in the P site, right? And then bacteria, the first codon is formal methionine. It's just a modified methionine, but that's a difference in prokaryotes and eukaryotes. It's methionine in eukaryotes and formal methionine in prokaryotes. So it binds in the P site, the next amino acid binds in the A site, and then, hold up, sorry. And then the ribosome moves down the mRNA in a 5' to 3' direction, right? And the ribosome also catalyzes the connection of these amino acids. Cool. All right, so here's some differences now. Transcription translation can be simultaneous. The start codon codes for formal methionine and mitochondria and chloroplasts. There are multiple ribosomes that can translate your mRNA at the same time. So that's what allows for quick gene expression, quick growth, all those good things. In eukaryotes, we process our RNA. We add a 5' cap and a poly A tail to the mRNA, and it exits the nucleus, and then it can be translated by ribosomes. But it needs to be capped and tailed so it can exit the nucleus. And so it'll be stable. And again, the start codon is for methionine, and one ribosome translates one strand of mRNA before translation begins again. And so that's different than prokaryotes. see. Yeah, and the chaperone proteins, that kind of makes sense. Like they're helping fold the amino acid into its 3D structure so that all of the amino acid chains can interact. And then there's a signal sequence that will tell the protein where to go. The protein must be removed. So like if there's a signal sequence for the cell membrane, the chaperones will direct it to the cell membrane and the signal sequence will be cleaved. And then in the endoplasmic reticulum, you can modify proteins by adding sugars and lipids in eukaryotic cells. Yes, so in eukaryotic cells, we have our poly-A tail and our 5-methylguanosine cap on the 5' end, and we splice out introns and put the exons together. And bacteria do not do that usually. Yeah, and you guys can look at that chart. Quorum sensing. So this is what allows bacteria to sense their own density. So it's like their way of talking to each other with their little inducer molecules that they make and they secrete into the environment, right? And so they can coordinate collective gene expression so that they all are coordinating a response at the same time. So yeah, first you have your signal synthesized, it diffuses. There are sensor molecules that bind to receptors on all the bacterial cells that are around, and then the receptor, once it receives a signal, whenever the signal reaches, I guess enough of a level, the receptor will bind to the promoter and stimulate transcription. RNA polymerase will transcribe genes involved in attachment, motility, biofilm formation, stress responses, vitamin synthesis, all the things that bacteria would want to do together as a group. Let's see. Signal transduction. Yeah, you'd have this is like with quorum sensing, you have a stimulus and a sensor protein and response regulators. So yeah, knowing that there are just sensory proteins in the cell membrane that facilitate responses from the outside of the cell to the inside of the cell, right? And it usually occurs through phosphorylation. Phosphate groups are really common in biochemical pathways for signal transduction. So we talked about like with glycolysis, glucose gets phosphorylated and then it can enter glycolysis. Cool beans. Yeah, so bacterial gene regulation. Constitutive means that it's always on. So these genes are always going to be transcribed and translated and expressed. And these are enzymes in the central metabolic pathways. So those enzymes that are involved in glycolysis and the PPP and the TCA cycle, all of those metabolic pathways that you need for survival to generate energy for the cell, right? Inducible, this would be like the lac operon, because you don't need to break down nutrients unless they are present, right? So then these operons are inducible in the presence of these metabolites. And repressible would be whenever, like for example, tryptophan. Bacteria also organize genes that synthesize amino acids in an operon. And so you always need amino acids. But once they reach a certain level, you don't need to waste your energy making that anymore, right? So operons that are responsible for synthesizing enzymes needed for amino acid synthesis, wow, that's a way to say it, are repressible because you can turn them off when you make enough of your amino acid, right? And he said it much better than me. Enzymes involved in building macromolecules, so anabolic. Alternative sigma factors. So here's our way to regulate transcription. Sigma factor, it recognizes specific promoters, right? So that's what it tells RNA polymerase where to transcribe. But cells can produce specialized sigma factors and anti-sigma factors to inhibit transcription, all through interacting with the promoter and RNA polymerase. Repressors block transcription. And yes, after the promoter, there's an operator sequence, and repressors bind to the operator. So even if RNA polymerase is bound to the promoter and ready to transcribe, it can't transcribe because the repressor is in the way. Activators positively stimulate transcription, which makes sense. And then a protein, so an activator would bind to an enhancer or an activator binding site that facilitates RNA polymerase binding to the promoter and upregulates transcription. Right. Yes, you need an inducer molecule to bind to the allosteric site. And the allosteric site, I believe, is just not the site where its main activity is, so not the part that interacts with the DNA. And then you get transcription when the inducer binds. So transcription is on, and you need a co-repressor. So sometimes you need a co-repressor in order to allow it to bind to the operator. Yes, and an inducer would bind to the activator, change its shape, and allow the activator to bind to the site. So I guess a big picture is that you have repressors and activators that need co-repressors and inducer molecules, right? All about signaling. And yeah, here's an example of the lac operon. And so normally, the repressor is bound to the operator if lactose is not present, right? You don't need to transcribe these genes that are involved in lactose metabolism. And when lactose is present, some of it's converted into a form called allolactose. And it binds to the repressor protein, and then that causes the shape of the repressor to change, so it doesn't bind to the operator anymore, and then polymerase can transcribe because lactose is present and you need to break it down. And this is a good point too, that glucose is the preferred source of energy for bacteria, right? So when glucose is present, the bacteria will first metabolize the glucose, and then once glucose is used up, then you say, oh, there's lactose present, so we should go ahead and transcribe this operon and metabolize the lactose, basically is what this is saying. Because lactose, and the way I think about this is lactose, it's a disaccharide of galactose and glucose, and it's broken down into its monosaccharides, glucose and galactose. So lactose is getting broken down into glucose. So it makes sense that if glucose is present, then you don't want to waste energy breaking down lactose into glucose when glucose is already there, right? Okay. Let's see. Some main pictures from this. Glucose transporter is a signal, so if there's high glucose, it'll get transported into the cell, and the unphosphorylated form of the glucose transporter indicates that glucose is still available. Okay, that means it's phosphorylated glucose, so it's an unphosphorylated form, right? Because this transporter is bringing in glucose and it's phosphorylating glucose. So if it's unphosphorylated, that means it's active in phosphorylating glucose. Yes. If there's low glucose, it will be in its phosphorylated form. So not much glucose is available. Right, so it has nothing to transfer its phosphate to. Inducer exclusion in high glucose. the unphosphorylated form of the transporter prevents the lactose transporter, okay, from functioning. Because lactose can't come into the cell, then you don't make allolactose, then you don't transcribe the lac operon, right? Because you have high glucose. And so this transporter component is not phosphorylated because it donated its phosphate to glucose. So it also, so it acts as a signal that, yeah, we have glucose. And then it also blocks the lactose transporter in because you have glucose. So there's no need to bring lactose in. But if it was phosphorylated, then it couldn't block this transporter, and then lactose could come in and signal, and then the lac operon would be transcribed so that you could break down the lactose. Yes. And again, I know he's not big on all of the enzymes, but if he highlights something on a PowerPoint, like I would go ahead and know that. So phosphoenol pyruvate is an energy source for phosphate transfer. And the enzyme E2A is what transfers the phosphate group from the transporter to the glucose. And these enzymes are hexokinase or glucokinase in eukaryotic cells. So yeah, knowing that glucose needs to be phosphorylated to enter glycolysis. And in bacteria, E2A is the enzyme that transfers the phosphate from the reporter protein to glucose. Yeah, and RNA interference. That's kind of crazy. In eukaryotes, they can synthesize short RNA sequences that interfere with gene expression. Yes, RNA-induced silencing complex assembles. The RNA in this complex recognizes the mRNA, the messenger RNA that would be translated into protein, and it targets it for destruction. And then enzymes chop up the RNA. Right, and it's all based on this. This enzyme is guided by the sequence in the short single-stranded RNA. Cool. So hopefully that helped. Let's see what else we got. With Sarah, we talked about chapter 6 a little bit. So I guess we can do carbon fixation and photosynthesis. You guys that have biochemistry on Friday probably know a lot more about this than me because it's been a while. But let's go for it. Yeah, chemolithoautotrophs and photoautotrophs use carbon dioxide to synthesize organic compounds, which we cannot do, right? We cannot fix carbon, but chemolithoautotrophs and photoautotrophs can. Carbon fixation, it requires a lot of energy, and the most abundant enzyme in the world, Rubisco, ribulose bisphosphate carboxylase. Yes, Rubisco. That's a very important enzyme for carbon fixation. And it's kind of fun to know that it's the most abundant in the world. So the Calvin cycle, carbon dioxide is added to ribulose 1,5-bisphosphate. It starts the cycle. And then you have an input of 12 ATP, a lot of ATP. And NADPH is also used. So you need 12 ATP and you need 12 NADPH. And it produces glyceraldehyde 3-phosphate, which you guys all know is used in biosynthesis. It can get shuffled to all the other pathways, glycolysis, pentose phosphate pathway. Whoa. Almost into the meeting. Okay. So you get 12 glyceraldehyde 3-phosphates, right? And one molecule of fructose 6-phosphate, and that can enter glycolysis as well, right? So glucose 6-phosphate is converted into fructose 6-phosphate. Every pathway is connected. Oh, this is great. Series of complex reactions. And then you need another 6 ATP to regenerate ribulose 1,5 bisphosphate. So adding another phosphate here to ribulose 5-phosphate. So you need to add one to the first carbon. So that's the Calvin cycle. A lot of ATP. So I guess looks like 18 total ATP required. Is it gonna let me go? Ah. Yes, intermediates from glycolysis and the PPP can feed into the Calvin cycle. So glyceraldehyde 3-phosphate, glycolysis, and the pentose phosphate pathway. So if we go back here, yeah, glyceraldehyde 3-phosphate can enter here, make some fructose 6-phosphate, and also be converted into ribulose 5-phosphate. Ribulose 5-phosphate from the PPP can feed in here, get converted into ribulose 1,5 bisphosphate. Start the cycle again. And NADPH from the PPP, yep, that can also feed in here. Calvin cycle energy investment. So one molecule of fructose 6-phosphate. 6 turns of the cycle. Wow. That's cool. Oh yeah, and you use 18 ATP and NADPH. But where does all this ATP come from? I'm not sure if this means one molecule of fructose 6-phosphate is made from 6 turns of the cycle. That would be something to look up, guys. Sorry, that's my bad. Right, so this ATP comes from photosynthesis. Sun, it's wonderful. Right, so they harvest energy from the sunlight and use it to power carbon fixation. So you've got your light reactions that uses sunlight and converts that to ATP, and then your light independent ones use ATP to synthesize the organic molecules. It's all about photosystems in the chlorophylls, bacterial chlorophylls. They have pigments that capture the light, and then it's all about electron transport. So these would be probably some good vocabulary words to know. The chlorophylls are the pigments in plants, algae, and cyanobacteria. Bacteriochlorophylls are used by anoxygenic bacteria. So those do not generate O2, oxygen. So yeah, usually, photosynthesis generates water, right? But anoxygenic, they do not generate oxygen. So we all, plants generate oxygen for us, but these anoxygenic bacteria do not generate O2. And then, let's see. Okay, bacterial chlorophylls allow bacteria to grow in habitats where these other photosynthetic organisms can't grow. Cool. And then you have, the accessory pigments absorb wavelengths, other different wavelengths of light. So the carotenoids and phycobilins. And another thing is this is all, I believe, in chapter 5 of our textbook. Photophosphorylation, using energy to fuel ATP synthesis and generate NADPH or NADH needed for carbon fixation. So this is the process of converting the sunlight into chemical energy, photophosphorylation. Yep, so the sunlight comes in, excites the reaction centers of the chlorophyll. It gets excited. There's electron carriers, again, proton pump that creates the gradient for ATP synthesis, more electron carriers. Well, oh, and photosynthesis system 2 is before photosystem 1. It's just back. One of them was discovered first, I think. Then sunlight excites photosystem 1. You get another excited chlorophyll electron carrier. And then you reduce NADP to NADPH. And then you can use that in biosynthetic reactions. One more. Fermentation. Not all prokaryotes have the electron transport chain, and it's said 3 times. So that probably means we should know that. So fermentation, it stops oxidizing glucose or other compounds completely, and it uses organic intermediates as terminal electron acceptors. The ATP comes from glycolysis, reducing power from glycolysis, and the electrons still go to a terminal acceptor, but it's not oxygen. My bad. Not it doesn't oxidize glucose completely. Right. Well, oxygen is the terminal electron acceptor in us. Yes. Yes. And in bacteria. Some of them. Okay. Whoa, it's been a week. So fermentation, we talked about how this converts sugar, starches, carbohydrates into alcohol and acids. So lactic acid fermentation is the one we've all heard about, right? Cool. So fermentation, if they don't have an electron transport chain, they'll use fermentation. If they lack an inorganic terminal electron acceptor, so like sulfur or nitrogen, those inorganic molecules that are not carbon, oxygen. And some microbes can switch depending on what's available, right? Cool. So this would be, yes. So organic carbon compounds and terminal electron acceptors, right? So it can be nitrates and nitrides, oxygen, terminal electron acceptors. So yeah, they can have different electron acceptors, but our terminal electron acceptor is always oxygen. Yes, in fermentation, pyruvate is a terminal electron acceptor. And you oxidize NADH to regenerate NAD that's needed to continue glycolysis. Yeah, and these end products are alcohols and acids. So ethanol is an alcohol, acid, butanediol, alcohol, lactic acid. You got it. Yeah. Whoa. He's all about history. Acetone used for World War I. I don't know if that's super needed to know. Acetone, butanol, and ethanol fermentation. Yeah, might just be a fun fact. Okay, yeah, and you can you don't have to use glucose. You can digest, like we talked about, polysaccharides and disaccharides, and those can enter these metabolic pathways. And then lipids, yeah, lipases separate glycerol from fatty acids. Proteins break down the peptide bonds. Basically, everything is recycled, and then you can still strip electrons away, right? So that would be oxidation. Oxidation is loss of electrons, reduction is gain of electrons. So you can still strip electrons away from these molecules and get energy from them. Lipid metabolism, beta oxidation, right? So you generate a bunch of acetyl-CoA that can go into the citric acid cycle, right? But you separate your fatty acids from glycerol through beta oxidation, and it's formed into acetyl-CoA. This takes place in the mitochondria. Let's see. Oh, and if you have a buildup of acetyl-CoA because you have a lot of fatty acids, then they can get converted into ketone bodies. And then that's what you hear about with diabetics, ketoacidosis, because they have a buildup of these ketone bodies that decrease the pH. Let's see here. Yeah, this is just showing how proteins, there's a loss of water, or they're hydrolyzed into their amino acids. So water is used to break the bonds. And then they're deaminated, the amino acids are deaminated. And then they can go into the various pathways, right? The deaminated portions. I think some of them can enter the TCA cycle. And then you can get some acetyl-CoA into the citric acid cycle. Acetyl-CoA, that's what gets you to the citric acid cycle, right? In inorganic fuels, chemolithoautotrophs use inorganic chemicals that are often byproducts of anaerobic respiration, right? So aerobic respiration is the presence of oxygen, anaerobic is without oxygen. So yeah, there's a terminal electron acceptor besides oxygen. And one bacteria's waste can be another nutrition source. Yeah, and here's some, these are kind of self-explanatory. Hydrogen, oxidized hydrogen gas. Sulfur oxidizes hydrogen sulfide. So HS, H2S. Iron bacteria oxidizes reduced forms of iron, and nitrifying bacteria oxidizes ammonia and nitrite. Let's see. Chemolithoautotrophs thrive in super-specific environments. They use inorganic carbon to power their anabolic reactions that build things, right? They extract electrons from these inorganic sources and pass them to the electron transport chain. Generate your proton gradient and energy for the cell. Okay. Yep, and here's a chart of that. Hey guys, I hope that helped. It was super fast, but I hope it helped you think of some questions that you might want to study more before your exam. You guys are going to do great. Email Dr. Daft or me if you have any other questions, and I wish you all the best of luck finishing this semester. Bye.